What is a recombinant protein? Well, it's the name given to a protein which has been made by usually getting a bacteria to uh, transcribe and translate a clone gene. I'm going to make the protein from that. The reason why it's called recombinant because it comes from recombinant DNA technology. Recombinant means recombine bits of DNA together that didn't belong together. You know, you get your bit of clone DNA and you combine it with the vector, with plasmid, or with the phage DNA. Okay, so that's where the word comes from. And right, why would we want to make cloned recombinant proteins? Well, let's think about some things which are, I think, insulin. Okay, insulin is used by type 1 diabetics on a daily basis. There's a huge amount of insulin worldwide made, and before about 1985, it all had to be purified from animals. And the problem with that was, well, one problem is that so bovine or cow insulin, which people use, isn't exactly the same as human insulin. It's slightly different, and after a while, the patients start to develop antibodies to it, and so they need more and more of it to have the same effect. So that would be one problem there. Let's think about another protein. So you think of the human growth factor. Right, so that's used for treating growth disorders. That is secreted by the pituitary gland in tiny, tiny amounts. And I believe that before recombinant human growth factor was available, it all came from the pituitary glands of human cadavers. And I think there was a problem there because to get enough for one dose, it's from hundreds and hundreds of people. And I think infectious diseases were spread by harvesting it in that way. So that's a problem there. There's other drugs. So you may have heard of granulate colony stimulating factor. Well, what does that? That makes neutrophils. It makes your bone marrow make neutrophils. Your white cells involved in fighting an infection. And levels of neutrophils in chemotherapy go down, and the patient can actually suffer life-threatening infection as a result of the levels of the neutrophils going down. So they're also given GCSF, and that stimulates the bone marrow to make more neutrophils during the chemotherapy. Another one would be interferon gamma. Interferon beta, sorry, that's a cytokine which is made in tiny, tiny amounts by white blood cells. GCSF is actually a cytokine as well. So you could never purify enough of that to actually give someone an injection of it. There's just not enough of it, and it's made in tiny, tiny amounts. So realistically, that's got to be synthesized by cloning methods. Interferon beta is used to treat multiple sclerosis patients. It slows down the progress of the disease. So there's a few different examples. So one more, EPO, which is a bit notorious. EPO is erythropoietin. And what that does, it's made by the kidneys. It's a hormone made by the kidneys. It stimulates the bone marrow to make more red cells. And so it's been used by professional cyclists and marathon runners. It's banned. They inject themselves with that to increase the number of red blood cells in circulation and make the oxygen carrying capacity better. It is also used to treat, because EPO is actually made by cells in the kidney, and people with chronic kidney disease don't make enough EPO, and they suffer from pretty bad anemia, and that is the proper therapeutic use for it. Again, you could never get enough of it from the natural source. It's got to be made by recombinant DNA technology. Okay, so now let's think about if you wanted to make some insulin. So here we have our plasmid. So you know our plasmid, normal plasmid, say PBR322, and here's your insulin gene. And you put the plasmid into E. coli. The problem is there that although the plasmid is the DNA for the plasmid is copied, so you can get lots of copies of the DNA. It's not translated, it's not transcribed, so it doesn't make mRNA from it, or translated, so you don't get any protein. So the E. coli will not make any insulin protein. So what has to be inserted into this vector is something called a promoter. Right, so this is a promoter site. That's a promoter sequence, and that binds RNA polymerase. Of course, RNA polymerase binds, and then that will make a messenger RNA of that gene. Now, the way, and the one that is very often used, there's lots of different ones, but very often one that is used is called the Lac Promoter. And the advantage of the Lac Promoter is it only works in the presence of lactose. So it's a sequence which occurs naturally in the E. coli chromosome. You know, you'll have the Lac Promoter will be in the chromosome somewhere, and it will be controlling it controls a few genes, but one of the genes it controls, it switches on, is the one that makes an enzyme which digests lactose. So the E. coli only needs that enzyme when a lactose is about, and that promoter only works in the presence of lactose. And that makes it quite useful for if you want to make insulin, because if you want to, you just want to grow the cells up and you want them to divide and so forth and not make insulin, then you just leave lactose out the culture medium. When you want it to make some insulin, then add the lactose and it will switch it on. So that's a very commonly used promoter. In addition to a promoter, sometimes, but not always, another sequence there, do it in purple, that's called a Terminator sequence. That's sometimes clothed in. Now, what Terminator sequence does, it stops translation of an mRNA. Sorry, it drops transcription, not translation, beg your pardon, transcription. Right, the RNA polymerase will bind to the promoter, and then, so that's the RNA polymerase. Then it will move around and transcribe the mRNA. And sometimes it can carry on past there and start transcribing all the other stuff that's not really wanted. So a terminator is a little sequence, and when it's in the RNA, it forms a hairpin. So it's sort of the piece of RNA doubles back on itself and base pairs with itself, and that causes the RNA polymerase to drop off the chromosome. Terminators are not always essential. Sometimes to get a decent clone protein, they don't have to be included, but promoters, yeah, you do always need to have some kind of promoter. Otherwise, the RNA polymerase will not bind to that bit of DNA, and it won't get transcribed. Okay, so in that way, you can make E. coli make lots of protein, well, quite a lot of protein anyway, and then it has to be purified from the E. coli, which is quite tricky, and proteins are not very robust molecules, and they tend to, denature, so it is tricky to do that. And also any drug which is a hormone, any drug which is so all of those ones, all those four we've got there, they all need to be injected, because obviously they're proteins, so they can't be taken orally, and they're all, they have to be kept in the fridge and things like that. You can't, they'll break down at room temperature. Now let's talk about some other proteins. Some need to be expressed in eukaryotic cells. And that's because usually the reason for that is because you get a protein. Here's the protein. And what happens, so a lot of eukaryotic proteins is they are glycosylated. That means carbohydrate residues are stuck onto the surface of the protein. So there's the protein, and then it will have a few carbohydrates at certain places. And those carbohydrates are pretty important to the shape of the protein. So, you know, if this is a hormone, it's not going to bind to its receptor if it is not glycosylated properly, because it'd be a different shape. Bacteria can't glycosylate proteins. It's only eukaryotic cells that can do that. So they can't be grown in bacteria. They have to be grown in a cell line, in a mammalian cell line, which is, mammalian cells are much more difficult to grow than bacteria, much more. The tissue culture medium is much more complicated and makes it a lot more expensive. And the mammalian cell line is usually one called CHO, which stands for Chinese hamster ovary cells. And the reason why they do come from a hamster, and the epithelial cells. And the reason why they use those is because of all mammalian cell lines, it's one of the easiest to grow. It's one of the toughest and can put up with sort of, not ideal conditions and still manage to grow. And so a lot of recombinant proteins are made in these, and that is much more difficult than growing in E. coli. The proteins which are made in that way are erythropoietin, that needs to be glycosylated, and also beta interferon, the one used in treating multiple sclerosis. So they can't be grown in a bacteria, they need to be grown in a mammalian cell line.